Well, hello everyone, and welcome to this Parkinson's Academy webinar in collaboration with the Cure Parkinson's Trust. My name is Diga Hastes, and I'm the head of the Parkinson's Academy and the chair for the session this lunchtime. Today, Simon Stutt, Director of Research at the Cure Parkinson's Trust, will be giving an overview of the charity, what does it do, and why is it different, followed by an update regarding ongoing clinical studies and what results we should be seeing this year. Mixed into this talk will be a short film. Following Simon's talk, Richard Wise, Director of Clinical Development at the Cure Parkinson's Trust, will join to answer your questions. Can I ask you to use the Q&A function for these questions? If you are unable to hear or see the presentation, I suggest you log out and then log back in again, because it's very rarely from our side that there's a problem. So, without any further delay, let me hand you over to Simon to start this session. Simon.
So firstly, I'd just like to thank the Parkinson's Academy for inviting us to speak. My name is Simon Stutt. I'm the Director of Research at Cure Parkinson's. Very briefly, a quick description of who we are. So we're a UK registered charity, but we have research projects going on all over the world. We fund internationally, both clinically and preclinical research focused on disease modification for Parkinson's. We were founded in 2005 by four gentlemen who had been diagnosed with Parkinson's. And while they were very pleased with the care that they were being given, they were a bit frustrated by the lack of research focused on actually curing the condition. And so they initiated this trust, the Cure Parkinson's Trust. And for the last 15 years, it's been focused on research driving search for a cure. Our mission is very basic. It is to identify, drive, and fund research with potential to cure Parkinson's. So we have a very narrow remit. And by cure, we mean any treatment that's going to prevent or slow down or stop, reverse Parkinson's, this neurodegenerative condition. And we do this with urgency, working alongside people with Parkinson's in everything we do. We're primarily focused around repurposing drugs, but have been shifting towards supporting new drugs as well. And we also do a lot of work in terms of measuring progression and stratification of the condition, particularly in the case of genetics. And preclinical, all the research that we fund has to be within 5 years of clinical translation. So again, we have a very narrow funding remit there. On the clinical side, our primary project is what we call the International Link Clinical Trials Program. And this is where an international committee gather for a 2-day face-to-face meeting every year at either the Van Andel Institute in Grand Rapids, Michigan, or at Cumberland Lodge in Windsor Castle here in the UK. And the committee is made up of 22 of the world's leading experts in Parkinson's. They are the best of the best. You have a lot of renowned researchers here, such as Professor Andrew Lee from the Institute of Neurology in London, Professor Michael Schwarzschild, who is the co-chair of the Parkinson Study Group in the US. I'm not going to go through them each individually, but these folks are world-renowned experts in the field of Parkinson's, and their job is to gather for a 2-day face-to-face meeting where they prioritize a set of dossiers that we've given them. And these dossiers contain everything that we know about a particular agent or treatment. And these dossiers are derived from a process which starts off with a literature review and discussions with biotech companies and researchers. And that allows us to generate a long list of agents that could potentially be dossiers. And we then evaluate the supporting data, looking at mechanisms of action, preclinical data, clinical data, and in particular safety. And that gives us a short list of agents. And from that short list of agents, we then generate a set of dossiers. We being Cure Parkinson's, we generate the dossiers in-house mainly for the purpose of maintaining style and format across the documents, just to make the reading and the intake of information easier for the committee members. These are all very busy people. And of these dossiers, we present 15 to 20 that we believe in-house have the best potential. And then at the meeting, before the meeting actually, there is a triage process. The committee members will individually go through the dossiers and decide which are the weaker dossiers. And that cuts it down to about 15 dossiers that are discussed during the 2-day face-to-face meeting. Each dossier is discussed for about half an hour. Also, during the meeting, we look at the progress and reports from ongoing clinical trials and other aspects of the work. And then ultimately, from the prioritization process of the dossiers, we get 2 to 5 that are prioritized. The dossier is quite a lengthy document. It starts off with a 2-page summary, which gives the committee member opportunities just to gather information at a quick glance. So there's information about the agent itself, the brand name or the owner, the IP history, the licensing status, the history of its involvement in the ILTC process. And then we look at the biology behind the agent, such as the pharmacokinetics, pharmacodynamics, the CNS penetrance, if that's a requirement, the efficacy in models of Parkinson's. But importantly, safety takes priority in this process, and safety and toxicology, mode of delivery, these sorts of things are also discussed on that first page summary. 2 pages in, we present a rationale for the agent. Why is this particular agent being presented? What is the hypothesis behind this particular dossier? And then from there on, it is an in-depth look at everything we know about the agent itself. So we go into the scientific background, what's the biological pathway being proposed here, the summary of the agent, what's its history of development, etc. And then there's the preclinical data and the clinical data. We also have been making a priority of exploring mechanisms of target engagement. How can we measure that the target specifically being looked at is being engaged with imaging or with biological assays? So that's basically the structure of the dossiers, and the dossiers will run typically up to about 20 pages each. How are the dossiers scored? Safety is primary, as I've sort of alluded to in this presentation thus far. The dossier is given a single score at the moment based on safety, scientific rationale, assessment of target engagement, brain penetrance, prior preclinical studies, and prior clinical research, and also relevance to people with Parkinson's. And this last piece is also an important item in the discussions that are held. The meeting is a closed door meeting, but what we do is we invite all the major stakeholders in the Parkinson's field into the meeting, and this includes people with Parkinson's. And there's a very dynamic discussion around the table. Only the committee members can vote and prioritize each of the dossiers, but they will turn, almost in every case, they will turn and ask the people in the room what they think about a particular aspect of the dossier that's being discussed. And there's a lot of expertise in the room, from a regulatory agencies such as NIH and NINDS, through to funding bodies such as Michael J. Fox, Silverstein Foundation, Parkinson's UK. And then, as I said, there's Parkinson's advocates also in the room. So a lot of people are invited to the meeting, and there's a lot of expertise there as well, expertise and experience. After the meeting, the top 5, typically the top 2 to 5 compounds will be moved forward. These will be considered prioritized. And then we go ahead, once prioritized, Cure Parkinson's is mandated to get these compounds into clinical trial. So then we start prioritizing trial sites. We go back and we talk to the stakeholders behind some of these dossiers. we look at funding opportunities, primarily through our ILCT funding partners. These are supporting partners who have signed on to help us fund these clinical trials over multi-year studies. But we also discuss with industry and with government agencies as well, potential for funding. And then we talk to regulators and key opinion leaders regarding trial design. And finally, we put an application forward to our independent research committee, and they will make a recommendation to the trustees of Cure Parkinson's as to whether the trial should be funded or not. And they do that with the support of independent expert reviews as well. So it's a lengthy process to set up these trials, but it's prudent, and it's worked thus far. So over the 10 years of ILCT, we've completed 13 trials of 9 prioritized drugs, and that's involved 1,300 people with Parkinson's. We currently have ongoing 13 trials that they are, have recruited 2,500 plus people with Parkinson's. And in the pipeline, there are 13 identified for trial, 13 drugs identified for trial, 10 of which have been prioritized by the committee. So it's quite a lot of work in the past, and a lot of ongoing activity around the International and Clinical Trials Program. And just to give you a visual representation of the program, this is what it looks like over time. You've got the years going across the top of the chart, of the graph here. And we're going to next go through some of these clinical trials and discuss some of the ongoing work, particularly on the dark blue ones where there's data going to be presented very shortly. So first of the updates regarding the ongoing clinical trials comes in the form of the GLP-1 receptor agonists. GLP-1 agonists are obviously a frontline treatment for diabetes, but numerous studies have demonstrated that these agents exhibit neuroprotective properties in models of Parkinson's. And just very recently, in some Cure Parkinson's funded epidemiological work, researchers have found that GLP-1 agonists are associated with a lower rate of Parkinson's in people with diabetes. So there is a very interesting association between diabetes and Parkinson's. If you have diabetes, you're more at risk of developing Parkinson's. And if you have Parkinson's and diabetes, you have a more progressive form of the condition. But this epidemiological data suggested that folks who were taking, people with diabetes who were taking GLP-1 agonists as part of their treatment for the condition were at a lower rate of developing Parkinson's. And a series of clinical trials has resulted in the publication in 2017 of a phase 2 clinical trial that looked at the GLP-1 agonist exenatide. There were 60 people with Parkinson's who were treated for 48 weeks, and then there was a 12-week washout period. And what the researchers found was that in the exenatide treated group, there was a reduction and then stabilization of motor score based on UPDRS Part 3. And while the placebo-treated group continued to progress in their Parkinson's. And at the end of the 48-week treatment period, during the washout, the exenatide-treated individuals started to progress back to baseline measures. And this work has been driven by Professor Tom Fultonay at UCL, pictured here alongside one of our co-founders, Tom Isaacs, the man, the myth, the legend. And Professor Fultonay has now got a phase 3 clinical trial started with support from ourselves. It's a phase 2 trial, excuse me, it's a 2-year trial looking at exenatide in 200 participants. And it's ongoing at the moment. It's now closed for recruitment. We should have the results mid 2024, hopefully. In addition to this, the initial exenatide studies have generated quite some enthusiasm in the Parkinson's field, both the community itself and the research community. And this has led to the initiation of additional GLP-1 agonist trials. One in particular is exenatide study being conducted in Stockholm. It's a phase 2, 18-month study involving 60 people. It's primarily focused around imaging modalities as endpoints, but that study is scheduled to complete towards the end of this year. In addition, there was the liraglutide study, which was conducted at Cedars-Sinai Medical Center in Los Angeles. It involved 37 people being treated with liraglutide and 18 people being randomly assigned to placebo for 54 weeks. The study hasn't been published yet, the results this is, but they have been presented. While there was no difference observed in UPDRS Part 3 motor scores, there appeared to be a slight placebo effect or placebo response on that measure. There were significant differences observed in non-motor features and activities of daily life and quality of life. So we were encouraged by these results, and they are adding to a growing pool of data supporting the idea that GLP-1 agonists could be a potentially new class of treatment for people with Parkinson's. And we're waiting on now the lixisenatide study. This is a phase 2 clinical trial that was conducted in France. 158 people were involved in the study who were treated for 12 months. And the results of that should be shared very shortly in the second half of 2022. In addition, there's a biotech company, Peptron, and they have generated a pegylated version of exenatide. It's a slow release form of the drug, and they have conducted a phase 2A double-blind placebo-controlled study in 100 people with Parkinson's. They were treated for 48 weeks, and that trial was scheduled to finish in the end of 2021. So hopefully we'll be hearing the results of that study this year as well. There's, in addition, all of the GLP-1 agonist studies that I've mentioned thus far have involved injectables, exenatide, liraglutide, and lixisenatide. They all have to be injected subcutaneously. And researchers have been working on new versions of GLP-1 agonists for the diabetes field that are oral. And the first one of these to clinic has been semaglutide. And currently there is a investigator-initiated phase 2 double-blind placebo-controlled study of 120 people with Parkinson's. They're being treated for 24 weeks in a blinded fashion, and then they'll be treated for a further 2 years in an open label fashion. And that study is expected to report at about the same time as the phase 3 study for exenatide. And it's quite encouraging that a lot of this research has stimulated biotech companies to get involved in this field. And the very good example of that is the Neurally study. So Neurally has developed its own form of exenatide, and they've conducted a phase 2 clinical trial involving 240 participants being treated for 36 weeks. And that study is scheduled to finish early next year. And so we should have the results at some point next year, maybe midway through next year. So a lot of really interesting data being generated from this field of GLP-1 research at the clinical level. And progress being progress, the diabetes field has been developing a new class of drugs, which are referred to as dual agonists. So these incorporate the GLP-1 receptor agonist, but they combine them with a what's called a GIP receptor agonist as well. This is glucose-dependent insulinotropic polypeptide. And the leading example, one of the leading examples here is a company called Kyrea. they have been developing a dual agonist, KP405, specifically for Parkinson's. There are other dual agonists being developed. The brain penetrance of those molecules is currently unknown, but in this particular case, this biotech company has been developing a dual agonist specifically for Parkinson's and other CNS indications. And it'll be interesting to see this move forward into clinical testing very soon. Another area of interest for us at Cure Parkinson's has been GCase activation. GCase is an enzyme, a lysosomal enzyme involved in the disposal of waste in cells. And about 10% of people with Parkinson's, it varies from region to region, but about 5 to 10%, depending on where you are in the world, of people with Parkinson's, have a genetic variant in the GBA gene. This is the gene that provides the instructions for the production of GCase enzyme. And researchers have been, what happens in these people, they have very low levels of GCase activity lysosomally. And what researchers have been doing is been looking for activators of GCase. And through a drug screen, the respiratory medication Ambroxol was identified as a potent activator of GCase, raising levels of GCase in cells. And this has been demonstrated also in vivo models of Parkinson's, where Ambroxol was found to rescue the models. So in 2016, this led to the initiation of a phase 2 clinical trial funded by Cure Parkinson's, Van Andel Institute, and John Back Foundation, some of our funding partners. And this study was, and a lot of the background research in this situation have been conducted by Professor Anthony Shapiro at UCL, Green Square. So the Ambroxol in disease modification for Parkinson's, or AMPD study, was initiated and involved 18 people being treated with Ambroxol on an open label fashion for 6 months. Baseline levels of serum and CSF GCase were measured, and at 6 months, the treatment was found to be very well tolerated. This is an extremely elevated dose of Ambroxol. It required the participants to take 21 pills a day, and Ambroxol is not the nicest of medications. It's got a very bitter aftertaste. So it was quite a heroic effort by all of the participants involved in the study. But most importantly, the study found that in all cases, levels of GCase were elevated as a result of this treatment, both in CSF and in serum. And one particularly interesting detail in the study is that regardless of genetic background, all of the participants had elevated levels of GCase. So if you can see on the graph on the left-hand side of the screen here, the white circles are people who carry a genetic defect for GBA, so genetic variant in the GBA gene, and the black dots are individuals who do not have a normal GBA gene. And in all cases, there was an elevation of GCase levels. So this sort of supports the idea that maybe GCase enhancers could be useful for not just people with GBA associated Parkinson's, there's GBA genetic variants, but also in idiopathic Parkinson's. And there's research, additional research supporting this idea, where scientists have looked at GCase levels over the normal aging of the human brain, and they found that GCase actually declines as we get older, particularly in areas that are vulnerable to Parkinson's, such as the substantia nigra, where the dopamine neurons are located, and also in the putamen, where the dopamine nigral cells release the majority of their dopamine. There's a general decline of GCase activity as we age. But of particular interest here is that people with idiopathic Parkinson's have a more reduced level of GCase activity in these particular areas of the brain. And we're not sure why this is. The biology of this is still being investigated, but it provides further support for the idea that individuals with idiopathic Parkinson's could benefit from some of these GCase activators like ambroxol that are currently being clinically tested. So this is an interesting, this is an area of interest for us. A third clinical trial that's going to be of interest going forward is our research on acetic oxycholic acid, or UDCA. UDCA is a bile acid that's naturally produced in the body, but it's used in the clinic for dissolving gallstones and for the treatment of liver disease. A few years back now, some researchers at the University of Sheffield identified that UDCA was neuroprotective in models of Parkinson's, particularly in genetic models. And they've published numerous studies now exploring this, as well as having independent researchers supporting their findings. And the UDCA particularly appears to be influencing the activity and the health of mitochondria. These are the small bean-shaped structures inside of cells that are in charge of energy production. They're the power stations of the cell. And problems with mitochondria are quite widely recognized as being associated with Parkinson's. And as I said, this research has been conducted at the University of Sheffield, firstly by Professor Heather Mortis-Boys. She did a lot of the preclinical work, and then Professor Oliver Bandman took over on the clinical side, and the UP study was initiated. This is a 48-week study of UDCA in Parkinson's. It's a phase 2 study, involved 30 people with Parkinson's, 20 were on UDCA and 10 were on placebo. And the results of that study should be announced very shortly. We're expecting them sort of over the next few months to be made available. In addition, there's been a lot of research in the past looking at the reduction of iron in people with Parkinson's. There is an accumulation of iron for some reason in vulnerable regions of the brain in PD. Iron accumulates with age, but in particular in Parkinson's, we see an accumulation of iron. Preclinical research has shown that iron chelators are neuroprotective in models of Parkinson's. And so there've been, this has led to a clinical trial that we initiated for the iron chelator deferiprone. This is a phase 2 clinical trial that's been conducted on 140 people with Parkinson's. They were treated twice daily for 9 months, and we are hoping to have the results announced in late 2020, so sort of towards the end of this year. So as you can see, it's quite an exciting period for Cure Parkinson's. We've got a lot of clinical trial results coming out, and hopefully there'll be some good news in there. In addition to all of this research, these research results that are going to be announced, we've got a lot of ongoing studies. And I just want to reference 2 of them here. Firstly, there is the azathioprine study. This is an immunosuppressive medication that's used for the treatment of inflammatory conditions like rheumatoid arthritis. There is currently a study being led by Dr. Caroline Williams-Gray at Cambridge University. It's called the AZA-PD study. It was started in 2020 in the middle of a pandemic, but it involves 12 months of treatment with azathioprine with a further 6-month follow-up of 60 participants. And that study, as I say, is ongoing at the moment. In addition, there is a neurotriptyline study, neurotriptyline being the tricyclic antidepressant. And there's a lot of preclinical research to suggest that tricyclic antidepressants have a neuroprotective effect in models of Parkinson's. And neurotriptyline was selected by the ILCT committee for clinical evaluation. And it is currently one of the drugs being tested in the ADAPT-PD study, or antidepressant trial in Parkinson's disease, which is recruiting 408 participants in 30 sites across the UK. And we are funding a sub-study of that, looking at motor progression in the participants who are taking neurotriptyline. Just before I finish, I wanted to point you towards one of our Parkinson's advocates, Dr. Kevin McFarthing. If you want more information about a lot of the ongoing clinical work, not just what we're doing, but also what's going on in the world of Parkinson's, I'd point you towards the HOPE list. This is a list of ongoing preclinical and clinical work focused on Parkinson's. Kevin has a background, he's a biochemist, and he has a background in biotech. He's spent his career in biotech developing therapies, so he's now using some of his skills to further help the field in terms of keeping track of where we are with ongoing research for Parkinson's. And we work with Kevin very closely. Each year we put out a clinical trial highlights article that reviews the ongoing clinical trial pipeline for Parkinson's disease therapies, both disease modifying and symptomatic. So if you're interested, by all means, please have a look. So just to summarize, Cure Parkinson's is a research charity that's focused on disease modification for Parkinson's. We fund both preclinically and clinical research. And we've had, we're in our 10th year of coordinating the International Link Clinical Trials Initiative. We have 13 completed trials, 13 ongoing trials, and we have a lot more in the pipeline about to get started, COVID willing. And highlights to look out for in 2022, the initiation of the phase 3 Ambroxol study, more GLP-1 receptor agonist clinical trial data coming. There's also the UDCA study that I mentioned, the UP study, that results are expected very shortly. And of course, the deferoprone ion chelator study results, they'll be coming out as well. And in addition, we'll be starting, hopefully, more clinical trials as well. So with that, I'd just like to say thank you very much. My contact details are here if you want to get hold of us. And I'm happy to share these slides. There's nothing confidential. And what I believe we're going to see now is a short video from involving one of our patrons, the rugby legend Mike Tindall, meeting some of our advocates. So I'll stop sharing and we can start that maybe.
Living with Parkinson's means a completely different way of life than it used to be.
My advice for anyone who's been recently diagnosed is, most importantly, don't be too hard on yourself.
Really important that people recognise you don't have Parkinson's, your family has it.
A good friend said to me, you may have Parkinson's, but Parkinson's has not got you.
So, Mike, the last time we saw you was at your mum and dad's house.
It was.
Are you wobbly today?
Yes, I'm wobbly
You're a
today.
bit wobbly.
It will be wobbly, yeah.
You all right?
Yeah, not bad though.
How's everything else? You jig jog? Been out for your run this morning?
But we're here today to talk to other people and families who are affected by Parkinson's. What do you hope to learn?
I just think it's all about the different stories, the different journeys. Everyone's symptoms are so different. I think the more you can learn and absorb, the more people it will help.
Ready to meet them?
I am ready.
Let's go.
My name is David. I'm 77 years old. I lived with Parkinson's for 14 years. Oh, that's better.
So you're 63 when you got diagnosed?
Exactly, yeah. What I had was symptoms were tingling in my left side. And I'd been a fairly active sportsman, not at your exalted level, but I played a lot of rugby for
Do
fun.
we need to talk about Wales-England rugby first? Or,
Get it out of the way.
No.
And I thought, oh, this is trapped nerve, it's just a problem. To a neurologist, within 2 minutes he said, you've got Parkinson's.
David came home. He'd gone to the hospital appointment on his own, and he came home and told me, I've got Parkinson's, and I laughed. I thought,
matter of fact,
yeah.
I've got Parkinson's.
I've got Parkinson's. We stood in the hall.
What are we doing for tea?
Yeah, exactly. What are you making? Because he's the cook in the house. So what are you making for tea? And I didn't, I didn't accept it. And then he got the message across. Actually, I have got Parkinson's. We're 14 years down the line now, and I think I'm probably still in denial quite a lot. And you kind of started living with it psychologically straight away, and I've been blocking it out for years.
My mum also is basically a primary carer and sort of puts her own things aside, which she shouldn't be doing. My mum's been amazed by the stuff that Sally did with my mum and dad, and how many people have now offered to come and have a chat, play chess with my dad, play cards with my dad while she can get out.
He doesn't sleep well at night.
Like your mum, we do put our own needs aside, and I focus on my husband, my children, and grandchildren now as well.
He's a husband. He's not your job.
Yeah.
So you've always got to make sure you get that balance of that right. Otherwise, it can weigh you down. I've seen it with my mum and dad. It can weigh you down.
My name is Omotola. I'm 41 years old. I got diagnosed with Parkinson's disease in 2016. I think it's because of my age, my race, and my gender. Nobody really thought about Parkinson's. With Mimi, I remember when she was much younger, and I used to braid her hair. It was more like a bonding experience for both of us. And as Parkinson's progressed and I lost my fine and gross motor skills, and I lost the ability to do that, and that was very difficult and challenging for me. I didn't want to fall into despair. I knew that I didn't want to let this illness get the best of me or be overcome by it. I've looked at this as an adventure. It's not an easy one, but yeah.
Wow.
My name's Alison. I'm 52 years old. I've been diagnosed with Parkinson's for 6 years. You kind of just feel like your whole world's been blown apart. I was probably in that dark place for 3 or 4 months. I remember crying every morning and every night. God, this is what I've got to deal with, and it's, and it's day in, day out. I think I kind of, as a mother, you kind of don't want to... You want to protect your kids a little bit, and there's no point in telling them the full story right at the beginning. And I was a nurse. I nursed for 20 years, so nursed lots of people with later stage Parkinson's, so I knew what it looked like. And that's all I could think about when I went home that day in the car was, oh my goodness, that's what my future looks like. I kind of knew I needed to do something positive, turn it into something positive, and for me that was raising awareness.
I'm Amapal. I'm 68 years old, and I've been living with Parkinson's for 11 years.
Do you want to just describe your journey of what you first thought about when you noticed you might be having symptoms, and then how was the journey to then getting to the doctor and getting checked?
Well, I was ignoring the symptoms, if I'm honest. The tremor was the first symptom. I was ignoring it because I thought, I can conquer this, it's nothing.
Belle, did you notice the tremor right at the start? Were you involved in the process, or did Amapal hide it from you as well?
I noticed the tremor almost immediately, but he's very stubborn, and he didn't want to go to the doctor for a long time until somebody else suggested that he ought to go.
She didn't take very long to tell me that I've got Parkinson's. It didn't dawn on me. I didn't accept it for the first 3 years or so. You think you're going to get on top of it, and it's nothing. It can't affect my lifestyle, but it does.
It's just such a wide spectrum of people. I sort of connect with David because I see what my mum and dad have gone through. And it's very similar age when they got diagnosed, very similar story, slower symptoms. Same with Amapal in terms of, he's sort of been figuring it out for himself, and it's been fascinating, actually.
So what needs to happen now?
Well, I think a few things have come out of today. I think there is a real place to be building a database that is easy to find for everyone to use, to go and people to share their stories, share their symptoms. Obviously, doctors, neurologists are going to differ in their advice, so you almost need to read the stories to actually figure out what's going to work for you, what's going to sit better with you. So I think that's definitely somewhere we could get to, that would a resource that people who are newly diagnosed, or if people who are walking the path and just at a plateau, can go and try and tap into and basically upskill themselves with it.
Well, where do we go from that? I see there are no questions coming in at the moment, Simon, so I have one for you. Going back to what you spoke about earlier, the research committee that you have at Cure Parkinson's Trust, can you quickly tell us who that's made up of?
Yes, so an important part of being part of the Association of Medical Research Charities is to be open, transparent, and completely unbiased. And so we have a completely independent research committee led by our chair, Professor Alistair Coles. Alistair is a research scientist from Cambridge University, but his speciality is, he's a clinician researcher, but his speciality is in the multiple sclerosis field. So he brings an independent perspective to the research. And then we have both clinicians and research scientists, as well as people with Parkinson's who have a background in research, also involved in the committee. They meet 4 times a year, and typically they will receive an application, they will review the application. If it is within remit and they think worthy of further investigation, they will send it out for peer review. And then the next meeting, which is 3 months later, they will take aboard the peer review comments and make a decision regarding the application, whether to recommend it for funding to the trustees or not. That's basically our process for funding research.
Excellent, Simon, thank you very much. And Daniel McDonald, a geriatrician, has just come through and said huge thank you to you for the great insight into the current research. It was amazing. So pat on the back there. Still no questions. So just some thoughts from both you and Richard. Are you hopeful? It looks like you are. Are you hopeful that a cure is on its way? I think it's very exciting that there are lots of potentially interesting results coming through this year. Even though we've been through a pandemic, things don't seem to have stopped that much. They've kind of carried on, and I think that's quite exciting. So what are your kind of gut general feelings? I'll go to Richard first, just to give Simon a moment of quiet. Richard, what do you think?
Well, we've had some good clinical results with the 3 GLP-1 trials we've done. Every one of those has shown benefit, either to motor or non-motor. We've got a phase 3 trial that's come out of that, so we'll see how that plays out. But we've also got another GLP-1 trial, which Simon talked about, lixisenatide in early stage Parkinson's patients, about 150. We're going to hear about the results in October, I would have thought, certainly this year. And I would be surprised if they don't, if that trial doesn't find some benefits as well. So, and then of course, there are a number of other GLP-1 trials in Korea, America, Sweden. I think it's a very promising target, and we're fortunate there are so many trials in that area now. People have looked over our shoulders over the years and thought, oh, that's a good idea. So they've taken a similar approach, and that's great for us because we're seeing the results of more trials than we originally envisaged. So I think a lot of the other biological targets that Simon mentioned earlier are extremely promising. interested to see what happens with iron chelation and Amboxol and GCase, and some of the others that he mentioned, and some that he didn't mention. I think, you know, you could say it's kind of more shots on goal, but I think we're trying to think through the biology perhaps more than a footballer thinks through the football.
Excellent. Simon, anything to add?
Yeah, I get really quite enthusiastic about it. But an important part of our job is to manage expectations, and what patients really hate to hear is that the cure is 5 years down the line, 5 years down the line, 5 years down the line. So what I like to say is that within my lifetime, we will see some sort of disease modification for PD, and that I'm confident of that simply from the background of the research that's currently being conducted. To put it all in context, in 1997 we had the first genetic risk factor for Parkinson's, which was variants in this gene called alpha-synuclein. And then the next year we had another one, which was Parkin. A couple of years later, LRRK2. And over the first 10 years of this century, researchers were looking at what the biology is, what's going on in the biology associated with these genetic variants. And in the second decade of the century, they've been developing the tools for manipulating and modulating this biology. And now we're at the point of where we're actually clinically testing these ideas. It's never a good time to have Parkinson's, but it's a really exciting time for Parkinson's research because we've got all this clinical activity going on based around this biology that's developed from the research of just thousands of researchers. And it's all culminating now in these key clinical trials that are ongoing. So in the next few years, we'll have certainly an idea as to whether our thinking and our hypotheses and theories are correct. And hopefully we'll be offering the community novel therapies to slow down or stop the progression of Parkinson's.
Brilliant. Well, Simon, a huge thank you to you. Thank you, Richard, for being around to answer questions. Thank you, everybody, for staying till the very end, and hope you have a great week. Cheers. Thank you. Bye-bye.
Thank you
very much.